Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. This is the sixth episode of Season 5 of Plantopia, and the third of a three-part series featuring 2025 APS Fellows. Today I welcome Dr. Carolyn Young, professor of plant pathology and department head for entomology and plant pathology at North Carolina State University in Raleigh, North Carolina. Dr. Young completed an NZCS from Central Institute of Technology in Wellington, New Zealand, as well as a BS, an MS in genetics, and a PhD in molecular biology from Massey University in New Zealand. She also completed a postdoctoral fellowship in the Department of Plant Pathology at The Ohio State University. In 2006, Dr. Young joined the Nobel Research Institute in Ardmore, Oklahoma, to lead the mycology and plant pathology program, working her way up to the ranks of full professor in 2019. In 2022, Dr. Young took on her current role as department head at NCSU. She is the first female head in the history of the department. Dr. Young is internationally recognized for her pioneering work on fungal grass endophytes, which encompasses endophyte discovery, genomic diversity, secondary metabolism, and their application to forage systems. At Nobel Research Institute, Dr. Young collaborated with agronomists, breeders, and ecologists to release improved cultivars and advance understanding of plant-fungal interactions. She has also contributed significantly to understanding the impacts of pecan scab and cotton root rot. Dr. Young has published over 140 publications, 2 patents, and released 2 tall fescue cultivars. She contributed to over 250 genome sequences of Epicloe, Venturia, and Phytophthora. Dr. Young has contributed significantly to APS, serving as the founding editor-in-chief of the Phytobiomes Journal, APS's first open-access journal, from 2016 through 2020. She has also served as senior editor for Phytofrontiers and participated in an APS task force on content delivery. With a legacy of innovative research, transformative mentorship, and leadership, I'm thrilled to welcome Dr. Young to Plantopia.
Thank you for having me. I'm really excited to be on the podcast, and the first time for me, for sure.
Absolutely. I wanted to start off by congratulating you on being elected a 2025 APS Fellow. As I mentioned in the 2 previous episodes with Dr. Widmer and Dr. Bain, fellows are recognized for distinguished contributions to the profession of plant pathology and one or more of several defined areas. So what did that feel like, receiving that email notification that you were a 2025 APS Fellow?
So it was actually a phone call.
Oh, it was?
Yeah, it was a phone call, and it was a phone call from Karen Garrett. And my first assumption was, oh, APS needs me to do something.
Yeah.
And I always love answering a call and saying, yeah, of course, I'd be more than willing to help with that. And it wasn't. It was, I was being awarded APS Fellow, and it really was this overwhelming feeling. I felt very humble with it, and I know the other recipients have felt the same as well. And when I've put packages together for other people, I never imagined that I would be one that would also have the potential to receive a fellow.
Yeah.
I really appreciate the opportunities and my involvement that I've had within APS. It's such an important organization for plant pathology.
Yeah.
It leads the way.
And it's clear that you're another member of the humble scientists that have been awarded an APS Fellow Award. So congratulations.
Yeah, thank you so much.
In what specific category are you being recognized?
I'm being recognized with my service to the Phytobiomes Journal, as I was the editor-in-chief that was set that journal in its path going forward, and also within my research career as well. And some of that is associated with plant pathology, but some of it's associated with beneficial symbionts, which are becoming more and more important in our agricultural setting.
Absolutely. It's easy to recognize that you're a New Zealander or a Kiwi, as some might pick up from your accent. You did your NZCS, which is your New Zealand Certificate of Science, your BS, your MS, and your PhD in New Zealand. And 3 degrees were at Massey University, and that's a pretty sizable university there in New Zealand. What got you interested in plants and plant pathology?
So my background's a little bit different than your average student. I'm a non-traditional student. I'm also first generation, my family, to go to university. So when I was finishing out high school, I really didn't know what I wanted to do. And university wasn't on the horizon for me because I didn't have anyone in my family suggesting or leading the way to show you that's what you would do. And I ended up going to a function at the high school, and there was a teacher from the Polytech, and a Polytech is like a vocational training organization, and they talked about the New Zealand Certificate in Science. And what that was, a certificate that you would get where you were doing some of the basic science knowledge, but they were teaching you how to be a technician. It was really an apprenticeship of being a lab technician. The way that I grew up as a kid, my father ran his own business. He was a butcher, and he had a lot of apprentices in his butcher shop, going through his butcher shop. So I understood what an apprentice was,
Right.
and I understood that I could go and work and I could learn as I was working. So that's actually what got me into science. I enjoyed chemistry, I enjoyed biology. They were my favorite subjects. And this kind of allowed me to go, oh, I could go and work at a university, which is what I did. But I hadn't thought about going to school there. So that was interesting. So my first job was to help with teaching the 300 level biochemistry teaching labs and setting those up and setting up those lab courses. And then during summer, we would get to work with research, and we'd work with different professors, would ask you to come and work in their lab. And I really enjoyed the research side of it.
What was your first research project you worked on?
I was working with a biochemist who was extracting compounds from some plants, from potatoes. And honestly, I don't actually remember the major details about it anymore. But what I can tell you is that when I actually joined a research lab as a full-time person, that was in the lab of Barry Scott. And I stayed in his lab for around 17 years. And that's when I did my bachelor's. I finished my New Zealand Certificate in Science. I did my bachelor's part-time, and then I did my master's part-time, and then I became a PhD student full-time in his lab. So he started off working with rhizobia and understanding nodulation genes and nodulation in rhizobia.
Mhm.
And then he brought in some fungal endophyte work and fungal secondary metabolism work. And he stopped the work on rhizobia, and we shifted completely to a fungal biology lab looking at these really interesting secondary metabolite pathways in some of the fungi, Penicillium paxilli, but also in these grass endophytes. And then his whole lab ended up looking more closely at that grass endophyte interaction and understanding the biology between those two partners.
Yeah, and we'll talk about epicloe here in a little bit. Want to get into that a little bit. I want to come back to this certificate of science. Is that like the equivalent of associate's degree?
Yeah, I think you could say that. And so it's interesting being at NC State now, we have what's called the Ag Institute, and that's a 2-year associate's degree. And you're supposed to, as part of it, you need to have some internships. So you need to get some work experience. And I'm a very visual learner, and many people are. And so having that experience where you're learning and then you're able to apply that knowledge is just so beneficial for people. And it is very much the same. And I equate our Ag Institute to the equivalent of what I got with my New Zealand Certificate in Science. Now, that degree doesn't exist anymore. That certificate doesn't exist anymore. I'm not sure what's in its place in New Zealand. There's always been these structural changes in academia over time, and things get absorbed. And I haven't followed through to see where it has ended up and how much of that still exists.
Yeah, it's interesting to me. I have an Associates of Applied Science, a 2-year degree. I also have a bachelor's, a master's, and a PhD. So like you, I had every step along the way. And I think it was really valuable for making mistakes and learning from making big mistakes at a smaller level so that when I started my PhD, I knew how to approach experimental design and things like that. I want to come back to the fact that you have 3 degrees from the same university in the same lab. And I'm a big fan of that. If you find a mentor that works for you, and particularly in your situation, you were working and doing school part time. But in New Zealand, Is there a push like there is in academia here in the United States where you have to diversify your degrees and go different places?
So New Zealand is a little bit different. We have 7 universities. That's for 5 million people, which is very unusual if you think about the number of universities that we have in the United States or even in a state. It's significantly more per population. So while there's some regionality, so people will often go to the university where they've grown up in the region. That's quite common. And sometimes you do stay, but there wasn't as strong a push for students to go out and they had to go to another university. There were student assistantships that were available and they would be advertised. So you would get people coming in from a new university or from a different university based on those assistantship that got advertised around New Zealand. But it wasn't an expectation. So it wasn't unusual that someone stayed on at the same university for all of their degrees.
Yeah.
But it was interesting when I came back to the US, when I came to the US and found out that, oh, it's really unusual for people to stay in the same university and that there was a bit of an expectation to go around and get some different experience.
And I think in recent years, there's been pushback against that. And I support that pushback because this idea that people have to uproot their lives and spend a ton of money to move across the country for 1 or 2 years of guaranteed income to move again, it's not sustainable. And being away from your family support network is really difficult too. I did my bachelor's and master's at the University of Maine, and I really clicked with my undergraduate advisor and was able to stay on there. And he did say that if you wanted to do a PhD, I'd like you to go elsewhere. But I think that it's come full circle. I think it's more common for people to stay put if they find a good thing because quality mentorship is hard to find. And if you have it, why would you walk away from it?
So I do want to comment about my supervisor, Barry Scott, because he was an incredible mentor to me. We had a really good working relationship in terms of I was his lab manager and I knew how he liked experiments to be done. And I remember sitting in his office and I would have some exciting results and I would be showing him the results and he always would say, but what do the controls look like? And so then I learned to flip the conversation and say, here are my controls.
Yeah.
Now, let me show you the experiment. And so that was really important that I got to teach the other students that were coming through. Don't forget your controls. I called him, he was very controlling, but in a need for controls way.
In an experimental design way.
Experimental design way. Yeah.
Yeah. You do your PhD. Well, you do several degrees under Barry Scott, and you move to the U.S. to the place often referred to as the New Zealand of America, Ohio, right? Where you worked on Phytopthora infestans.
Yeah.
Was it always an interest for you to come to the U.S. to continue your scientific journey?
Yeah, so back to that idea that I did my PhD or I did all my studies in one place, I made a decision that if I was going to do my PhD and stay on in Barry's lab, then I really needed to leave New Zealand. And I had a family, I had a husband, and I still have a husband and 2 kids. And so we were looking for a place where we actually could afford to live on a postdoc salary because we would be taking a big cut in pay. My husband would have to give up his job and a big cut in pay to come to any new place. And moving a family across the world is a big ask, especially we had a 12-year-old and a 2-year-old when we made that decision to move. So I was looking for an environment where we could afford to live, but I was also looking for somebody that I could connect with in the same way that I had this amazing supervisor in New Zealand, who would be another amazing supervisor. And I met up with Sophie and Carmoon at a couple of conferences before I decided to move. And we had a number of conversations and I had so many positive comments made about him. He was very up and coming at the time. When I joined his lab, it was right before the RXLR effectors had been identified. And how I had it described to me, the Phytophthora community all would come together every year and they were starting all of this genome sequencing and gene expression studies. And they were looking at their genome sequences and their favorite genes, which were effectors. And they did some alignments and they realized there were these conserved domains, the RXLR and the DER domain in these proteins. And so when I arrived, I didn't actually know what I was going to be working on specifically. And Sophia gave me the project of trying to identify as many RXLR effectors and Phytophthora infestans as possible and start doing some functional genomics with them. So I made a library of these RXLR effectors from different isolates so that we could see the diversity that was within these effectors and maybe how they functioned. And then there were some R genes that we would co-express into tobacco. And that was the beginning of the project. And it was a lot of fun to be working with a new organism and understanding the differences between this endophytic system that I'd been working with and a plant pathogen and how plant pathogens work.
Yeah.
So that was a really exciting side of things. Sophia is an incredible researcher, and the way that he thinks and the way that he ran his lab and how people were organized and the way that the projects were run was really integrative. And so I was working with one grad student in particular, and that was really interesting to me because I needed someone to show me. I didn't know how to do this. I'd never done this before. So I was happy to let me be your hands for a while. I was getting my project set up and getting my libraries made. And so that was really fun to explore and learn a new system in that sense.
And then also you came along at a time where, like you said, you had all these genomic resources and you were on that big P. infestans genome paper. Is such a pioneering paper. And I feel like that was probably like the golden age of Phytophthora discovery. And similarly, when I talked to Min Gallagly, who was a faculty member here, talking about the discovery of the sexual state, it's like there's these like bursts of productivity and bursts of understanding of this important pathosystem, and you were part of one of those bursts.
Yeah, which being part of a burst like that was really an incredible opportunity, and the excitement around it. And being part of a community, that's what we are in science. We're all part of these incredible communities where we're engaging with each other. What is it? There's only 2 degrees of separation between anyone in a science field, because somebody who knows that person, and that's it. So those connections and how the whole Phytophthora community was growing and developing, and they were all sharing ideas. They were all sharing either these R genes or the AVR genes, and it was just a really exciting time.
I don't want to get into all the details of it, but I can't help but think that your participation or your involvement in that community at that time set the stage for you to be the department chair at NC State University, because there's a lot of great work on Phytophthora there, no?
I hadn't thought about it that way, but I did meet Jean Rostino at that stage, and that was pretty exciting. And just seeing a female scientist that was so incredibly successful, and it's such a pleasure working with her now in the department. I've got a few department connection stories with a few of my faculty that I can elucidate.
So you finish your postdoc and then you move on to your 16 years at the Nobel Research Institute in Oklahoma. Do you want to talk a little bit about the work that you were leading there as a principal scientist?
So first of all, let me explain what the Nobel Research Institute was, and it is now. It's changed a little bit since I left. So this was a research institute it's based in small town Oklahoma, in Ardmore, Oklahoma. And it was a phenomenal research institute. Think about a large plant science department where you've got the basic science that's going on, you've got translational science that's going on, and you've got the extension that's associated with it. And it was really like this big organization, and we had a more centralized focus. So our focus was around beef cattle production was a really big component of it. But cows eat grass, and so cows eat plants. And so what are those plant systems how can we improve them? How can we improve the agricultural side of things, the agronomy side of things? So as I said, it was the basic science where you had in the plant biology department, you had a group of scientists that were doing all of this leading plant science research, understanding what makes plants tick. And then you had us, I was in the forage improvement division. We were taking some of that information, but we were also trying to translate our science. And mine was around endophytes in grasses and forage grasses, translate that science working with the breeders. We had the breeders within our group and then the agronomists as well. So how do you take this premier breeding material and get it into the hands of farmers? And then there was this extension component that also worked with us and we worked alongside. So it was an incredible organization that was dedicated to helping farmers in our region. Initially, it was this 100-mile radius from Ardmore, which encompassed some of Texas and up to about Oklahoma City. And then really it was more about the whole state, and now it's really opened up into the whole country and beyond. But Noble's gone through a different change. They're now working with regenerative agriculture, and they're really more focused on soil improvement, but not having the major inputs and utilizing cattle for this, or grazing animals to enhance the regenerative nature of the land. And so they don't have that same desire to have the leading plant biology and the plant breeders and things like that. So there was a change in the organization. But back to Noble, back to what I did. When I saw the ad, it was about grass endophytes. And it was about working with a grass breeder. And we take, so I tell you the story about tall fescue. It's a really important grass. It's a forage grass. It's a turf grass. It's widely planted across a lot of the United States. It's very prominent in the eastern United States. And it is a very important grass for grazing animals. Now, the problem with it is that there's a fungal endophyte that's in this grass, and that fungus produces toxins that are toxic to the grazing livestock. And that can lower animal production, their average daily gain. So their productions are lower, and it can cause fertility problems. It can cause some lameness, and they can lose, there's vasoconstriction that occurs, and they can lose their hooves and the switch of their tail and things like that. So not great for grazing animals, but it helps protect the grass, provides drought tolerance, insect protection. So it's got all these really huge benefits, and it was really instrumental in the 1940s during the Dust Bowl to actually get plants in the ground. It's a perennial crop as well. And I've talked to farmers, especially in around the Kentucky area, that have told me that their father, and this sort of ages up people, but their father just speaks really highly of tall fescue because they wouldn't have had anything to feed any animals in the time of the Dust Bowl had it not been for the introduction of fescue. How do you get rid of toxicity? And that's where my program came in. And there's actually a group in New Zealand. I worked with them, or I worked alongside them or collaborated with them when I was in New Zealand, but I haven't worked for the company specifically. And they had identified endophyte strains that you can use the same species, but they don't have the toxicity, the livestock toxicity. So this endophyte seed transmitted, gets distributed in the seed. And so I needed to work really closely with our grass breeders to be able to introduce the endophyte or make sure a lot of tracking that we do have endophyte infected plants and that we had the right strain that we were working with. It's really easy when you're collecting samples from fields and you're using harvesters, that seed lots, there can be some mixes and things like that, or carryover from seed lots. And so we had developed a really nice quality assurance pipeline that they could use for that to make sure we never carried through any toxic fescue.
And these were mostly ergot alkaloids, or these are like indole type terpenes?
So yeah, that's a great question. So these are the ergot alkaloids, are the ones that cause the fescue toxin. And the vasoconstriction is one of the key symptoms and a key problem associated with it. Cattle are very susceptible for when they're ingested, and that lowers it. Think about just not feeling great, and I'm not going to eat as much, and there's a bit of a malaise going on there. Thoroughbreds can cause a lot of early abortions in horses if they're on toxic fescue. So that's a huge problem, as you can imagine, a huge problem out in Kentucky, where fescue is thought to not have originated because it's come over from Europe, but where it really became part of a dominant part of the agriculture, or dominant part of the land and spread out further from there.
Are these alkaloids like swainzanine and the ones that cause the Slobber's diseases, or am I thinking a different one?
So it's a different one. It's another interesting secondary metabolite that's produced by an endophyte, and those ones are in legumes. Scientists that I get to collaborate a lot with are those that are looking at endophytes and fungi that are producing secondary metabolites that may be toxic to grazing animals. And so swainzanine is another one of those compounds. It's still a fungus, and then a legume, a dicot versus a monocot. But some relationships, the genes are all found in gene clusters. So that makes it easy when you find one gene associated with a secondary metabolite pathway, you tend to find them all because they're all closely associated together within the genome.
2 doors down from me is Dan Panachone, and he's a mycologist who works on ergot alkaloids here at WVU. I don't know if you saw his recent work on Paraglandula clandestina, which is a new culturable Paraglandula species from Ipomea tricolor.
They have a number of similar types of genes, including the ergot alkaloid genes. They have indole diterpene biosynthesis genes as well. We've collaborated with Dan. I met him when he came and did a sabbatical in New Zealand. And
Yeah, he had mentioned that.
yeah, so there's these small circles. Yeah.
You've worked on an apicloe. Like, what is it about these clavicipitaceous endophytes that you find so intriguing and interesting?
So if we look at grasses all around the world, around 30% of the cool season grasses are infected with an epicloe species.
Wow, that's a
And
lot.
yeah, and one of my fun research projects was looking at the USDA grid system and asking for the cool season grasses, for these collections of cool season grasses to be sent to my lab so that I could determine which lines were endophyte infected and which ones were endophyte free. The markers we use for quality assurance for the tall fescue work actually work really broadly across all of the epicloe, and we can go in, and we went in and screened a large number of the material within the USDA system to determine which ones were endophyte infected, what were the alkaloids they were able to produce. So there's a number of other classes. We've mentioned ergot alkaloids, there's the indole diterpenes, there's the pyrrolizidines and the pyropyrazines. So they're also known as the lolines and pyramine. And these secondary metabolite compounds have anti-insect properties. Some of them have anti-mammalian properties. But there's also these endophytes are very diverse, and so are their secondary metabolite pathways. And so we could unfold the natural diversity, discover the natural diversity that these endophytes have. And now we've got to go back and put the ecological component to it. So we know what they are likely to produce or do produce in terms of the alkaloid production. And now we've got to go back in and say, what's the ecological advantage of that? And so
Right.
there's some secondary metabolite pathways that they don't have the complete pathway, and there's common genes that have been mutated, but they're independent mutations, and that's suggestive that there's an ecological reason for stopping a pathway at a certain point. We just don't know what that is. And that's going to be the exciting component of it.
Absolutely. I recently dipped my toe into the clavicipitaceae, working on Conoidea luterostrata, which is an intimal pathogenic fungus. And running it through anti-smash, it has twice the number of biosynthetic gene clusters that Claviceps purpureae has, and some of these metarhiziums. And it's just, wow, there's just a world of biosynthetic potential in that genome. And then you start asking questions using mass spectrometry to see what the secondary metabolites are and which ones are produced in culture. And it's just so fun.
Yeah. Now, what's really interesting with the grass endophytes is they don't produce these metabolites in culture. They need to be in the host plant to produce them. With your work, Matt, you might want to go and look and see what's actually being produced when it's on the insect as well.
Oh, unfortunately, the insect stage that we're looking at, they're so small, you'd have to collect a lot of biomass. But I noticed in looking at your CV that you co-authored a recent paper in Global Change Biology on how warming disrupts plant fungal endophyte symbioses, particularly in leaf tissue. What does that tell us about the fate of 30% of grasses under a changing climate?
Yeah, you're right. What does that climate changing climate going to do? And some of this work stemmed from some samples that we sent to the University of Kentucky, where Rebecca McCulley has a set of heaters buried in the ground so that you can grow plants under a changing environment and increase the temperature of the soil, so you can put them under a couple of degrees higher growth rate. And then they also had a reduction, or try to mimic what the environmental conditions are going to be rain-wise. Not so much that it's going to be a constant drought, but that when you do get rain, it's all going to come at maybe just a couple of times a year. It's going to be very intense, and then there's going to be nothing there. And so understanding how these endophytes, the role that they may have. Is there a fitness advantage to the plant having an endophyte, or is there actually a fitness cost as well? I was part of a collaboration with that. So I had a fairly minor role, but some of our material was what was going in there, and it was some of the native grasses. So I have a real interest in native grasses and the epicloes that are associated within those native grasses, and the discovery and diversity associated with them.
But I think it speaks to a larger issue and a larger question about resilience in plant communities and the role that phytobiomes and endophytes and mycorrhizal associations, these certainly provide benefit even in extreme conditions, and maybe more so in extreme conditions. But what if things get more extreme too quickly? What's the tipping point there?
I'm not sure that we have a full answer on that. We never have a full answer. We're always trying to search and find what is the ultimate. Just to bring up that phytobiomes side of things here.
Sure.
And so for the audience, what a phytobiome is, we're looking at all of the microbes that are associated within a plant system. And what was really interesting for my transition into thinking about just a single fungus that is a dominant species in a host, to maybe this broader community of microbes that are there, but also then the environmental conditions that may be associated. It's not just what in the soil or on the plants it's not just the plant, right? There's a whole community of everything that's there. And so how our science has changed over the years. And I saw a really great publication figure that shows how sequencing has changed and how that's influenced how we now are able to do our studies.
You were the editor-in-chief at Phytobiomes from 2016 to 2020. Here it is 2025. We're like, in the last 10 years, phytobiomes has become such a buzzword in plant pathology. And certainly we're understanding the role that the phytobiome plays in ensuring sustainable global food security. And then that's one of our big motivations. If we can grow crops better with less inputs, or just a better understanding of the microbial communities, then that's going to be beneficial for humanity. But what else do you think we've learned in the last 10 years about phytobiomes that may be surprising or unexpected? Do you think it's, like you were explaining, do you think it's really shifted the way we look at plants and plant communities?
I really do think it has shifted how we look at plants and plant communities. And there's been a number of scientists who were very strictly involved with the plant pathogen system, but as our ability to understand and look more broadly at communities, they've realized that there is a bigger, wider story within at a phytobiomes level, because there are these associations that we never considered. We only ever considered a plant and, for my case, an endophyte, or a plant and a pathogen directly. And so now that we have these additional tools, such as the high throughput sequencing and expression studies and the metabolomics that we can do, we can actually build a much bigger picture of what's really going on in a plant system and how that system is able to be improved. Can we make synthetic communities to enhance the growth of plants and reduce our inputs, for example? I gave examples of epicloe being interesting because it makes its own insecticide compounds.
Right.
So it's protecting its host against insects based on the production of secondary metabolites that it's making. And so being able to understand all of these dynamics within the phytobiome context is becoming more and more important, but also there's just so much that you're learning. There's so many more organisms out there that we've never looked at or considered because we didn't have a way of understanding them. And you gave an example of Dan Panashone now with a periglandula that can be grown in culture. All the previous periglandulas were scraped off leaves because they don't grow in culture. But that's an example of just how our understanding is able to be improved based on the tools that we have at our hands now.
I like the fact, too, that we can look into the assembled genome and start to understand some of those interactions. Like you were saying with the insecticide, at some point in time, it was used to combat, and maybe still to this day, is used as an anti-feedant or something like that. I think about how a number of members of the Claviciptaceae produce antibiotics. And if you're in an insect host and you're competing with bacteria for that substrate, you need to keep bacteria out. So you're getting a glimpse into maybe the bacterial community that's fighting for that real estate.
Yeah. So it's becoming more of an ecology type question, right? Where I'm a molecular biologist. And so I'm very cut and dry with, I have my genes, I have my genomes and understanding that. But to what point? And it becomes more of an ecology question as to what's really happening. And that's phytobiomes is part of that as well. So what are the ecological reasons or advantages for certain associations? I always think about it as an advantage because I work with symbionts and beneficials.
So you currently head the Department of Entomology and Plant Pathology at NC State, yet you yourself have identified as a molecular mycologist before you just said you're a molecular biologist. I see you've joined APS and MSA in the same year, 2006. By the way, that's the year I joined APS. So I ask you here publicly, like, what's your preferred hat, plant pathologist or mycologist, or do you prefer not to claim loyalty to one or the other?
I've liked both a lot.
Yeah.
I do a lot more now with APS. So I can definitely say that APS is a very important society for our department, right? Because we half the department are plant pathologists, and I'm here to support that and the plant pathology discipline. As a fungal biologist, I'm always going to be excited about the MSA as well, the Mycological Society of America. And I did go to a large number of the fungal biology meetings, but they're all intertwined. There's maybe some projects that I work on that's more perhaps appropriate for MSA versus other projects that were more appropriate for APS. But some of the community members are the same. And that's part of the cool thing about science is the scientific community and the people that you get to meet along the way. And some years it may be at the MSA meeting and some years it may be at the APS meeting.
I think you're motivating me to come up with a Venn diagram of MSA members, APS members to see how many numbers overlap.
I'm sure
Yeah.
it's a lot.
Yeah, so it's really interesting. I want to come back a little bit to your time in Oklahoma. You worked on a number of things there. Obviously, you talked a lot about your endophyte work. I see that you published a number of papers, especially in the last 5 to 10 years, on Venturia, including the peach scab pathogen as well as the pecan scab. When I look up where those crops are most prevalent, Oklahoma is third in pecan production, behind Texas and Georgia. Peaches in Georgia, not necessarily Oklahoma. Did that motivate some of your work because regionally they were important issues?
So regionally, pecans is a really important issue where pecans are America's native nut. Big shout out for pecans right now. Okay?
Nice.
You don't go for these other nuts. Pecans are our nuts.
And the
meat
pie.
And they make a great pie. They make a great pie. And I love having a pecan pie on my slides as I'm giving a presentation. So they are a really important crop for Oklahoma. And to put it into context, as you drive around Oklahoma, we had 4 pecan trees in our backyard.
Wow.
So now, they weren't great producers. 2019 was a great year for pecans, and we still had some in our freezer. But that's how it's just so visible on the landscape through Oklahoma and Texas. And so that meant it was a really important crop. We get enough rainfall that the diseases in pecan, in particular scab is the number 1 disease, was really important. So the further west you go, the lower the rainfall and the less scab problems there are. But as you come out through Texas and then once you get into Georgia, it can be a huge problem just through the humidity.
Yeah.
So I was really excited about working and picking up a project that related to something that was native to the area. I mentioned native grasses as well. I really loved looking at the endophytes in native grasses.
Yeah. I definitely can appreciate the fact that throughout your career, you've maintained those two areas of emphasis. You've been devoted to the fungal endophytes and that work, and then starting with your Phytophthora work at Ohio, and then moving to Oklahoma, you've kept one of your feet in the plant pathology realm. So
Yeah.
that's great, and I think that positioned you well for your current role. And I kind of like to transition into that, if you don't mind. Clearly, you brought up the fact that in 2021, the Nobel Research Institute kind of transitioned from plant science to regenerative agriculture. Certainly, that seems like it was a motivating factor in that. One of the things I noticed and it jumped out at me on your CV is that in 2019, 2020, you did a tremendous amount of leadership training. And I wondered if the writing was on the wall and you were looking for your next challenge and you knew you wanted to do leadership. It worked out that way.
That's a really interesting observation. And Noble, as it was transitioning, they took us as scientists, as the senior scientists, and they did want to give us additional leadership training. They saw the value in that.
Mhm.
And then they opened some of it a little bit more widely to the broader scientists, the broader group at Noble. It wasn't something that I think I would have thought of doing myself hadn't the door not been open first.
Right.
And so you learn some stuff along the way. You learn how different people are and how we think differently. We may have the same motivation moving forward to, or the same desires, the same types of outcomes, but how we may approach getting there will be really different. And so we have to consider everybody in the room and how they may be thinking. And it's not just the people who talk the loudest or talk the most, right? Or first to jump in. We've got to stop and what is that thoughtful person who maybe isn't able to jump into that conversation. So it teaches you a lot about people and about interactions and things like that. And I think it was a stepping stone. Had there not been a change at Noble, I may not have thought about applying for a department head position, but I had some people reach out to me and say, hey, these positions are being advertised. It was a great time for, if you wanted to be a leader in plant pathology, there's been a lot of change in the plant pathology leaders over the last 5 years. It was an opportunity and I didn't think I could make that shift because I hadn't been in academia. I had been in a research institute for the last 16 years.
But on that note, like I did notice you had titles like assistant, associate, and full. Like they had professor level, basically ranks there at your institute. So it looked a lot like an academic institution in the way that it was structured.
Yeah, so in that sense, we were a research academic institute. We're a little bit of a cross between academia and industry in terms of there was an expectation to create products and have systems in place. We didn't have to teach, but I had a really strong desire for outreach and I had a great connection with the vocational training school next door that's called Southern Tech. And we brought in high school students. We developed a program to bring the high school students in to do internships within Noble. And so they got to work in the various research labs. So on that side of things, and on the extension side, outreach to farmers as well. So in that sense, it was like it, just not understanding all of the academic side of a department.
Yeah, I saw that one of your outreach things was you had grass screenings for endophytes for farmers, which is cool. That's kind of, we have a plant diagnostic clinic here at a lot of land grant universities, and it's really nice to see those services and see the value to the people that come in to drop off samples. They have a pressing problem and they need an answer. And similarly, if you have grass breeders or farmers and they're trying to figure out if they have organoalkaloid producing strains or cultivars of grass, they need to know, and you're providing that. I can't help but think when you mentioned the Southern Tech, is that what you said?
Yes.
That's almost like a nod back to your science certificate in New Zealand.
Yes.
It's the same thing. It's like you're paying it forward.
Yeah, exactly. That's exactly what it was. There was an opportunity. My friend, Dr. Fiona McAllister, she had been a postdoc at Noble, but then became a teacher, and then she led the biotechnology program at Southern Tech. And her and I got together. She's originally from Australia, so we speak the same language, right? We understood each other and we got together and hatched a plan about how great it would be to have these students be able to come in and do some internships. So they would have a kind of a capstone at the end of their second year. So it was a 2-year program. So when they were seniors, and they would have an opportunity to work in a lab. It was a minimum of 6 weeks. Most of the students got kept on longer, and most of the students would get a position over the summer as well once that had happened. So it was a really great opportunity. And a lot of these kids that came through the program, a lot of these students, sorry, that came through the program, it was a little bit like me. They didn't have family members that could advise them on going to university, or they didn't have that structure where they understood what their next steps could be. And so it was a wonderful opportunity to meet with students and give some guidance and help them navigate their path forward in terms of their future studies. It was really exciting to see the program grow. It was great for the postdocs because they were the ones that were often looking after these students and engaging with the students. So that gave the postdocs some mentoring experience as well.
Yeah, and that's critical. What a wonderful program. We're rapidly approaching the end of our time together, but I wanted to first talk about one more thing. And as a department head, it's like capacity funds and extramural opportunities dry up in real time, even if just temporarily. How do we retain and reassure early career scientists who are worried about rapidly shifting job market that to stay the course and that investing in a university education is still worthwhile?
Yeah, I sent an email out to my faculty with what the budget looked like, the proposed budget, and it's not what you want to see as a department head and trying to imagine what the future could look like, or how we have to navigate that future if we're not able to fund some of the programs that we have taken for granted, or that if we're a land grant university, right? So a large role is extension and helping growers. How do we maintain all of those programs?
I saw a proposed budget line for 2026 where the hatch funds were zero.
Yeah,
And
yeah,
and I know a lot of land grants depend on hatch funds, and without which you can't really function.
yeah. Yeah, so if that goes through as is, let's just say I don't wanna be a dean right now, um,
Yeah. Yeah.
because those are gonna be really hard decisions, but, they're gonna be hard decisions that as a college, you're gonna have to come together and figure out what those opportunities are. What other funding opportunities are out there, because a lot are drying up, and what would be sustainable in future funding. I really appreciate Bill Gates putting more money back into international agriculture. People like that, those types of funding opportunities
Yeah.
are going to be more valuable in the future as well. And while industry partners are there, if everybody's drawing on them, then they could be drained as well. And so how do we balance all of this? What are the needs? And how do we help our early career, our students and our faculty look for those types of jobs or opportunities, funding opportunities in the future? And it's a really important component of it. For the students, I think it's important that we acknowledge that not everybody wants to be an academic. I think when I was growing up, that was if you're going to do a PhD, it's because you wanted to be an academic. These days, we have a large number of students that come through and they are really interested in the science, but academia is not the career path they want to go down. They're looking at other opportunities in industry, and being in North Carolina, we have a huge ag industry here, so it's a great place to be with the various companies, the ASF as an example. So we've got great representation of these companies. The nonprofits, communications, a lot of the students are coming through, and communication, science communication is a really valuable area, or an area that they really want to expand on. And I think that's going to be critical for our future moving forward.
No doubt. No.
Yeah, yeah. Like maybe we're really good at telling our science to a scientific conference and we get a lot of joy out of that, but we've got to tell our stories to the public in a way that they want to hear and understand. And we're trying to do that within our own department, and thinking about the messaging that's coming through our social media about the value and the impact that our research, our extension, our education has. And that type of communication is going to be really important going forward. We should all be able to tell our family members, our people in the community about the value of the science that we're doing in a way that's engaging, and understand where our food comes from and understands what we do to keep that food production going and safe.
I think the other thing that we need to push back against, like, I'm at an R1 research institution here at WVU, and they really prioritize with PhDs over master's degrees. Yet a lot of these jobs you can do with the master's education, and there's still value in that, but it's always been undervalued. And I think universities need to reconsider how they value master's degrees, because it can give you the extra training beyond your bachelor's to get you qualified for a position. And you can earn a livable wage with a master's, not have to do the extra 2 or 3 years for the PhD.
I actually see a lot of value in our master's degree and master's program as a whole, and it does give you a really good foundation to be able to move forward. We have a number of people going from with a master's into extension agent programs, and so they're the people that are going to be at the face of the community. And I think in those sense, at somebody with a master's, they're going to get a slightly higher salary based on that, especially if they move into an extension agent role. So I really do see a lot of value in it. I see value in people being able to upskill, and that we don't all have to go through a traditional route for
Right.
education. And I think I'm an example of that, right? Where I worked full time, admittedly at a university, so it was easy to get that additional education, but it was because I was interested and curious.
Absolutely. Thank you so much for your time, Dr. Young. I really enjoyed talking with you today.
I appreciate it. It was a real pleasure to be here, and thank you for the opportunity.
We've just heard from Dr. Carolyn Young, professor of plant pathology and department head for entomology and plant pathology at North Carolina State University in Raleigh, North Carolina. Be sure to check out the show notes for Dr. Young's episode immediately following the episode description. I'm Matt Kasson, host of Plantopia. Thanks for listening.